This is the second lecture in the series on group differences analyses, looking at t-test and ANOVA. This one will be focused on the t-test particularly. So say Dr. Williams wants to do a study, and she wants to know if the two groups are different, and her sample's pretty small. Now, if she's got two groups, then her degrees of freedom is going to be a capital N. That's the total number of people in the study minus 2 because she has two groups. So if she has 26 people in the experimental group, let's say the ketamine group we've been talking about, and 26 people in the standard group, the Prozac group, then her degrees of freedom would be 50, right? Because she has 52 people total, a capital N, and N minus 2. And this is how you would do degrees of freedom for a t-test. You would write it out as you see there with the t and then the degrees of freedom in parentheses, which is 50. And then I just made up a number there, 4.3, let's pretend, is the actual t value. So this kind of gets you a sense of like where we're going, what we're doing, and tying it back to the degrees of freedom we were just talking about in the previous video. So the next thing we really need to think about whenever doing a t-test is to think about whether we have a between subjects design or a within subjects design. Now, if you remember back to experimental psychology, a between subjects design would be where everyone in the study gets only one level of the independent variable, or generally people are randomly assigned to one group or the other. What that means is like people get either ketamine or Prozac. They don't get both, okay? A within subjects design, what happens in that is everyone in the study gets every level, typically at different times. So in this case, they might get Prozac for a while, then they go off it, and then they would get ketamine for a while and go off it, and they would be looking at their happiness ratings when they're on each of the groups. So you have this two different ways of handling the data, but that when you have the different design, that means you have to do something different with the t-test that you're running. So one of the things you need to really know right up front is, am I doing a between subjects design, which is the most common one to do? People most commonly will assign people to one group or the other. But sometimes people do within subjects designs, and they'll, for instance, have people spend some time in treatment and have them spend some time in no treatment. And that actually is a within subjects design right there because everybody's getting a little bit of both. So this is just a brain break because all graduate students, they need brain breaks now and then. And it's just one of my more favorite of all of the Ph.D. comics that exist out there. This one is just really funny about how your week goes, what's happening on TV on Thursday, consider quitting on Wednesday nights, happy hour on Friday night, so you have a pretty low work output there. Weekly meeting with advisor causes you to just plummet because your ego is crushed on Thursday night, right? So you have all these kinds of little things that affect the amount of work output that happens during the course of a week. I just think this is just one of the funnier ones that they have. All right, back to work. So, assumptions of the t-test. One of the assumptions in a t-test is that you are going to have one categorical independent variable and that variable will only have two categories, right? Male and female, or Prozac and ketamine, or treatment and no treatment, you know. So those are your assumptions on this, and you must have that. The other is that you have one continuous dependent variable. So you're working on a continuum there. In general, t-test is fairly robust to violations and assumptions in general and can be a little bit liberal on most of them. And I'll tell you which ones you can't be. But this is just an assumption of how it's set up. Like if you run data that's not like this, you're running the wrong analysis on the data. One of the assumptions is unequal N. This can be a problem if you have a small sample, particularly where you have particularly wildly unequal N between your groups. So if in the male group, for instance, you have 25 participants and in the female group you have 250 participants, you have a serious unequal N problem at that point. You have too many people in one group or the other, and you're going to have to fix it. And you can look in textbooks about how to fix that. There are a variety of ways. But again, if you have a very large sample and you don't have that unequal N, if you have, 50 people in one group and 52 in the other, people just usually keep going and don't do anything about it. But some people are more strict and they would lop off those 2 people so that they have exactly 50 in each group. So it depends on how strict you need to be, and that is a larger issue in terms of what you're trying to do with your research and how well you're doing with some of the other assumptions too. Another thing that's an assumption in the t-test is that those bell curves for each group are normal, right? That you have nice, normal, pretty mesokurtic bell curves generally. This is also important for a small sample or low power. It's not that important for moderate to large sample sizes. But you would be looking at skewness for your groups to try to determine whether or not you have a normality problem. But how strict you are, whether you kind of go, well, -1 to 1 is okay, or whether you go, no, I need -0.5 to 0.5 as my standard on skewness, would probably depend on the size of your sample. So we're going to use a power analysis to decide for that. And a power analysis in a t-test is a pretty set thing. I ran this through something called G-Power. You can go online and find G-Power. It's a really nice program. It's free to download, and it's in English as well as other languages, and it allows you to do simple power analyses by just plug and chug. You just put in the information you have about your research study, and it will tell you a priori, how many participants you would need to get a certain power level, or it will tell you after the study is over and you put in how many participants you had and everything, what your power actually was. So if you expect a large effect, 50 should be enough for a t-test. If medium effect, you need about 100. If you're trying to detect something that actually has a small effect, right, it doesn't really affect people that much, you're going to need a very large sample for any kind of study you're doing, by the way. But it's like 600-ish is expected at that point. So just to be aware, it pays, particularly in a dissertation, to study things with large effects to medium effect sizes in previous research. So that's how you know what your effect size is what previous research has tended to show for that area of study. All right, so another thing that we assume is homogeneity of variance. So remember I showed you the 2 bell curves of the ketamine and the Prozac, right? And then we looked at them in different ways. And each of the ones that I showed you before were kind of the same. They were either kind of all bunched up or they were all spread out platykurtic, both of them, right? But if you have a small sample, especially, and you have one group that's mesokurtic and one group that's platykurtic, or worse, you have one platykurtic and one leptokurtic group, then you can be violating the homogeneity of variance assumption, right? This is, again, less important as the sample gets larger. You'll notice that theme throughout the assumptions. If you have a large enough sample, most of your assumptions don't matter. I cover assumptions in good detail for doctoral students largely because many of your dissertations don't have large samples, and so you really do have to pay close attention to the assumptions in that case. So you see a picture here of these 2 populations, and you can see how they have pretty different variances. The top one has a pretty small variance, and the bottom has kind of a medium to medium-large variance. And so if you have that, you have some concerns. Independent samples is one of those design issues, meaning if you are doing an independent samples t-test, then you better not have people getting ketamine and Prozac in your study, all right? Or you better not have people who are, in your treatment and they're also getting another treatment because they're talking to the people in the waiting room and they're like reading books about the other treatment. So it's really important for between subjects designs that your samples are actually independent. You didn't accidentally switch the rats in your rat maze study or whatever. One of the things that is something to actually watch for t-tests and ANOVAs too, by the way, is if you're violating multiple assumptions. And in particular, there's 2 assumptions that go together and make for sad graduate students that you see there. All right, and that is if you violate homogeneity of variance, which remember is a Levene's test, and then you also have unequal n, so your 2 groups are pretty different sizes, even if you have a moderate, decent power and you have a moderate sample size, we start to get really concerned about whether the data might have errors in it to a point where we can't trust our outcome. So this is one we will be looking at in our lab, and you should be looking at it in any research that you do. If they violate both of those things, this makes for sad graduate students. Now, in my labs, you're probably going to just keep going. You're going to report it and keep going. But if these things were really a problem and you had homogeneity of variance problem, Levine's test was significant and it was violated, and then you had wildly unequal n, you might actually have to stop the study, throw out all your data, and start over again with a new sample. So pray that that doesn't happen to you in your real life research world. All right, this is a bit of a self-test on what we've covered so far. Make sure you understand what exactly a t-test is doing. Most of that is from my first lecture. So we're talking about within group and between group variances, that kind of thing. What exactly is a degree of freedom? What does that mean when I look at that number? Can you come up with another example study that would be appropriate for a t-test besides comparing ketamine and Prozac? Can you give an example of what heterogeneity of variance and unequal sample size would look like? And where would you find that on SPSS output? And then any further questions that you might have. So this is the end of lecture number 2 on ANOVA.